How's everybody doing this morning? Any issue? Was everybody able to get into all the different parts of the textbook and connect? Or still having difficulty with the cadaver dissection tool part? Anybody? So far, so good.
Seemed to be okay.
Okay, glad to hear it.
I was just a little slow getting all set up. I should have been paying closer attention to the instructions, but we're all good now.
Very good. All right, well, looks like it's 8. We're missing a few folks, but hopefully they'll log in pretty quick here. We're going to run through chapter 2, which is pretty much, if you've ever taken a cell biology class, it's pretty much all of the cell biology class in one lecture. We're going to come back and revisit a lot of this information over the course of the semester as we cover specific different parts of the body and how things work. But this is just kind of a general overview of the cell primarily and kind of how the cell works within the body to allow things to get in and out of the cell. And we'll talk about kind of the different roles of some of the major organelles within the cell. So that's kind of the focus of today. I sent out a message last night about what we're going to do in lab. So we've got the ability in Zoom, if you're not familiar with it, of creating what are called breakout rooms. And so the breakout rooms, basically, I'm just going to be able to decide how many rooms I want based on how many people we have. And it's going to just kind of create smaller discussions that groups where we might have like 3 people or so in each group. And I'm going to have you work through a few of the handouts that are basically looking at a lot of the images that we showed last time. And those are the things that are going to be on the quiz coming up next week. So it's kind of a good way to at least help you guys kind of think through it together a little bit and get ready for the quiz a little bit. Normally we would do this in person, and that interactive part and sharing and learning is a little bit easier to do than trying to do it virtually like this. But that's what we have at the moment. So that's what we're going to try. We'll see how it goes. I haven't done, like I say, this class in this format. So some of the things that we've done in the past may or may not transition over quite as well to the Zoom model. And I'm not sure, maybe at some point this semester we'll get actually on campus and be able to do some more of these things in person and actually help you see and handle some of the models that we have. But if not, then we're just going to have to do it the way that we're doing it here for now. So let's, got one more person to let in, and I'm going to go ahead and share my screen and try and get rolling here. All right, I need to see if I can move some stuff around so I can see the other images that I kind of need to see, which is you guys. Don't know that this is going to work the way I wanted it to work. So I'm going to kind of go back to how I had to have it visually here. I was hoping to eliminate this clutter on the left-hand side that has all the slide deck images. But every time I try and enlarge this, it shoots it over to my other screen, and then I lose the chat box and everything else that I need to kind of help keep track and manage stuff in the class. So I'm going to probably just show these this way. Like I said, I was hoping to make it a little bit bigger and a little bit more clear for everybody, but it doesn't seem like it wants to let me do that. All right, so now I think I have everything where I need it. I can see everybody's smiling faces. I can see if people are coming in or out of the meetings and all that stuff. So let me back up just a sec. Actually, I guess we'll start right here. So again, we're going to go over all the cell stuff. the term, the prefix cyto just means cell. So anytime you see cyto, you should be thinking that it has to do with the cell in some way. So cytology, study of cells. Cells are itty bitty things for the most part. We do have some cells in the body that are pretty big, and so we'll look at a kind of a graphical representation of kind of how these different cells kind of relate. But most of our cells are going to be really small. You can't see them with the naked eye. We need some sort of amplification where we can magnify it and see things a little bit easier. We do have lots of different shapes. You know, the classic cell is always kind of pictured as being a little sphere, but it does depend on where that cell is located and kind of what its function is. All right, so this picture here is the one that kind of shows the gamut of how big a lot of our different cells might be. We talked last time about some of the different component pieces, how we need to have 2 or more atoms to make a molecule, 2 or more molecules to make tissues, things like that, and work our way through. And so all these cells are going to be components within these different tissues, within these different organs, and then within the person as a whole. When we look at the whole body. So this is running from 0.1 nanometers, which is itty bitty, all the way up to 10 meters. Now they show the person. This is not that this person is actually a giant. If we follow that line over to the side, it's showing the 1 meter mark, and it's a, you know, if somebody's 6 foot, 6'4", 5, they're going to be about 2 meters high. Most of us are going to be, I'm like 1.7 meters tall on 5'7". So that's obviously not a single cell. There's lots and lots of cells in there. But if you look right below that 1 meter mark, somewhere between 1/10 of a meter and a meter, and kind of on the upper end of that, we've got some of the muscle cells and some of our nerve cells are actually that long. When we look at the way that we're going to activate muscle in the body, we've got a neuron up here that's going to control and send a message through a long axon that's going to go down our spinal cord. And so depending on how tall we are, and let's say we're trying to activate a muscle that makes our toe bend, makes our toe flex, that first neuron that's in charge of making that toe movement happen needs to ultimately connect with the muscle fibers that are going to contract to do that. And so that the neuron up here is going to have a very long axon that goes all the way down your spinal cord, and then it's going to synapse or connect and communicate with another neuron. Now that what's called a motor neuron in the spinal cord is then going to send its long axon all the way down our foot, all the way down the back of our leg, through our calf, and all the way down to our foot, so that it can actually connect and send the message that's going to make those muscle fibers fire. So in that case, we've got 2 cells, those 2 neurons, one going from the brain all the way down the spinal cord, and then one going from the spinal cord all the way down our leg, that are one individual cell. And so each of those could potentially be, you know, 3 feet long. You know, if you look at somebody that's 6 feet tall, that's not out of the realm of possibility that those cells might be that long. If we look at some of our bigger muscle groups, like muscle in our thigh and our quadricep or our hamstring, similarly, we're going to have individual muscle cells that kind of run the length of all that whole contractile part of the muscle. You know, we've got tendons kind of on either end of the bone, but in between that, we've got muscle fibers that, you know, many thousands of muscle fibers that make up the whole muscle. But each individual muscle fiber is its own cell. And so those can also be pretty long, a couple feet long. Now, those are kind of the exception. Most of our cells are going to be much smaller. This one kind of in the middle of the diagram, this oocyte, that's basically the egg cell for that the woman's going to ovulate. So that's a pretty big cell in the body, but even that is much smaller than 1 millimeter. So it's closer to 100 micrometers, or like 1/10 of a millimeter. So it's pretty small. And then we've got a few other structures that we're looking at there. And we'll talk about a lot of these things too. So lots of different cell sizes that we might potentially see. So in order to visualize most of these cells, we need to use a microscope. And so we're going to look at different images over the course of the semester sorry, I had to let somebody back in. that show us different magnifications. And we've got the next slide is going to kind of show what these slides look like. So the light microscope is what you may have used in a previous biology class, where you prepare your little slide, put it under there, turn on the light. You've usually got some kind of, usually 2 or 3 options of magnifications that you can do, maybe 10 times, 100 times, 500 times, or something like that. And so it has limited ability to magnify things a lot, and it has limited ability to be really, really clear. So those are kind of our most crude way to use a microscope. The next two, the transmission one and the scanning one, both use electrons to help increase the magnification and improve the detail of the images a lot. So let's look at kind of what those images might look like. So kind of in order, this is our light microscope. This is our scanning electron microscope, or excuse me, our transmission electron microscope, and then this one is our scanning one. And so they're all looking at the same structure. We've got, in this light microscope, it's magnified 720 times, and they're trying to focus on the cilia, or the little hair-like projections that are kind of coming off of the border of that cell. With this transmission electron microscope, we're able to increase that magnification in this image up to 50,000 times its normal size. And so we get a lot more clarity of the image, a lot more detail. It's a lot more of a close-up. But it still is a flat 2D image. So both of these first two are just flat images. So they lack, it makes it a little bit easier to get depth perception and things like that with it. Our scanning electron microscope, again, this is also looking at cilia, but it gives you the ability to look at things kind of 3-dimensionally, and gives you a much greater appreciation of kind of how things are oriented to one another, and how it really might look, and how in this type of image, I think, really can help in understanding the function of these different components, because it just gives us a lot more context to the image than just looking at some of these flat images. So again, as you're reading through the book, as you're going through homework, things like that, you're going to see some of these images, and kind of pay attention to the little notation in the bottom corner about what type of image it is and how many times it's been magnified, and it should give you a better sense of kind of what you're actually looking at with that. All right, so let's talk about cells and kind of what they do for us. Each of our different cells is going to have one or more kind of key roles that it's going to help with. And this list that we're going to look at really doesn't cover everything by any means. So if we look at these functional categories, we've got a lot of cells that are coverings. So if we think about our own skin, our skin has got tons and tons of layers of cells. And that's good because it's going to kind of make them more expendable. Our skin serves a lot of protective roles. And so if we just had a single layer of cells covering our body, anytime we bumped up against something, all of a sudden we'd have a hole in us and we'd start bleeding and leaking stuff out. And so we don't want that. We want to have these, it's helpful to have those multiple layers of those skin cells to give us some extra protection and keep us more viable. So we shed those things constantly. We make new skin cells constantly. And that's why you always hear about like how gross like bed linens and pillows and stuff like that can get, because we're just constantly dumping all these cells out there, and those are just, they used to be a living part of us and now they're dead. And like, you know, bed bugs and dust mites and all those kind of things love to feed on those things. So wash your sheets often if I haven't convinced you of that with that lovely description. If we look over to the right, we've got some cells that are going to serve to connect things. And so that's just, when we talk about connective tissues, that's the cell types that are going to kind of hold us together. And so like underneath our skin, we have fascia, and fascia is just like a sheet of connective tissue that's going to kind of help in compartmentalize different muscles and different areas in the body. We also have connections that, like an ACL ligament, an elbow ligament that's going to kind of help keep our humerus attached to our ulna, tendons that attach the contraction part of the muscle belly to the bone where it links in. So those are all examples of kind of connective roles that cells might play. Now we've got a lot of cells that line things. And if you think about like your digestive tract, um, if we're talking about our intestine, as we get into the intestine, we need to have a lot of different cells that can produce secretions to help lubricate things and protect some of those linings from things like digestive enzymes that might erode them. We also need to have cells that line those intestines that are going to help us absorb the nutrients that we have consumed. So we have different types of cells that serve specific roles in that lining. Again, some that might secrete stuff, some that might help bring things into the body and get it into our bloodstream by absorbing nutrients. And so those lining cells are really important also. When we look at the immune system, we've got cells that are going to help protect us against invading organisms. So our lymphocytes are a specific type of white blood cells. We've got a lot of different kind of subtypes within that category, but those are things that are going to help our body identify and attack viruses, bacteria, things like that that might be getting into our body that we need to neutralize so they don't do further damage to us. So defense is a big role for some cell types also. Now we've got storage. And so the two examples that they give over here in the fat cells and in liver cells. So anytime we eat excess calories, regardless of whether those calories are coming from fats, carbohydrates, or proteins, if we don't need them right away for the body's use, our body's like, well, we might need that later. Let's put it into storage. And so if we have a carbohydrate and we've eaten too much carbohydrates and we don't need those right now, the body's going to convert that over into a fat. And then we're going to store it in these fat cells. And then if we're starving later and we don't have any food available, our body's like, oh, I know where I still have some of that stuff we could use. So we'll be able to break it out of the cell and change its shape and convert it back to a carbohydrate if that's what we need it for, or keep it as a fat if we want to burn the fat as energy also. So we do have storage roles. Nervous system, communication is a big part. And so I talked about those two different neurons in the brain and spinal cord that are going to help us control muscle function. And so we need to be able to send that message. I think about what I want to move. I've got to be able to convert that thought over into action and communicating through those axons that connect to the subsequent neurons in the chain are what are going to kind of help us do that. Our muscle cells are going to help with movement, and that's true with both skeletal muscle as well as if we think about our organs. A lot of our digestive organs also create movement that's going to help basically mix food up if we're talking about the stomach by contracting the stomach muscles to kind of churn things around a bit and help break them apart, mix in all the digestive enzymes with that substance that we've consumed to kind of break it apart. And then same thing once we get to the intestines. Our intestines also are going to be contracting in a couple different directions to kind of further help pulverize up and mix up all that food so we can get it into small enough pieces that we can then absorb it. And then we've got reproduction as kind of our last little category over here. And so our cells are going to go through the process of meiosis to create the gametes that then can merge to cause fertilization and start new life. So those are just kind of some of the primary roles that different cell types can play in the body. So let's kind of look at just a generic cell and look at some common features that most of our cells are going to have. And then we'll kind of dive into each of these areas a little bit deeper. So this term plasma membrane, sometimes it's called a phospholipid bilayer is kind of the more specific way to describe it. And we'll look at a picture that shows that bilayer membrane as well. So it's permeable, but it's selective. So some things can pass through that membrane pretty easily. Other things might need a special passageway to get in. And then some things we don't really ever want to let back into the cell. And so we want to block the ability of those things to get in. So it has the ability to kind of be discriminating about what do we want in and how much of that stuff do we want to come in the cell. And if we didn't have that membrane, then our cells would be much more vulnerable to damage. Things that we don't want in there would get in there and cause more problems for us. The cytoplasm just refers to kind of everything that's inside the cell except for the nucleus. So we'll go through all the different organelle types here in a little bit. But we've got this kind of, depending on where the cell is, it's usually described as kind of a gelatinous material that makes up the cytosol. So that's kind of more the liquidy component, but it's not like water. It's usually, like, say, a little bit more gel. Like in some cells, you're going to have less water to it. If we think about something like a bone cell versus a muscle cell, the muscle cell is going to have a lot more of water content than the bone cell will typically have. So that will vary depending on where the cell is, how much of that liquidy stuff there is, and how firm or viscous that cytosol is going to be. Inclusions includes a lot of little molecules, proteins, and carbohydrates and fats that the cell might need for later use. And then the organelles, just like the organs in our body, are specific structures in the cell that have very distinct roles. Our stomach doesn't do what our intestines do, just like the Golgi apparatus in a cell doesn't do the same thing that a mitochondria does. We kind of need all those different cell organelles to help the overall cell work. And so if we're missing one or more of those, or one or more of those components has an issue, then the whole cell is going to have an issue. Just like if our stomach's not working right, we're going to have, our whole body's going to be affected by that. And the nucleus is the so-called brains of the cell. So that's what's going to kind of control and direct the activity of the cell. When it needs to make more things, when it needs to repair more things, when it needs to absorb more things, it's going to kind of help manage all those things. So that's the CPU of the cell, so to speak. So this is kind of, you probably have seen images similar to this in high school biology, or if you've taken college classes anywhere else. There really is no cell that looks like this. This is just drawn to kind of include everything that we're going to talk about. The main thing that we won't find on a lot of cells is this long extension right here, the flagellum. The only human cell that has a flagellum is a sperm cell. So, and this is not a sperm cell, but it is shown on this image. So we're going to go through all of these structures individually, but this little purple membrane that we see, that's that phospholipid bilayer. And then we have all these different organelles that are located within it. And then the kind of fluid medium that surrounds it is that cytosol. So the cytoplasm is everything inside this boundary of that plasma membrane except for the nucleus. The nucleus kind of has its own membrane that surrounds it, and it's kind of considered separately from the rest of the things in the cytoplasm. So we'll kind of start going through these different parts and talk about how they work and what their roles are. So our plasma membrane, again, is a double layered boundary that's mostly those phospholipid molecules. And so we have what's called a phosphate head that's kind of spherical, and then it has two what are called lipid tails. So basically just two chains of fatty molecules that are kind of holding it together. And those individual phospholipid elements have a distinct charge to them. And so the phosphate heads are basically facing out on either side of the cell, either between the cell and the fluid medium around the cell, or between the cytosol and the cell itself. So those phosphate parts are kind of the perimeters, and then all the fatty tails line up together in the middle. And so we'll look at an image and we'll talk about that a little bit more. And so this is really the key to helping control what comes in or leaves the cell. Now we've got a lot of things that are embedded in that membrane that are also going to help with controlling things coming in and out. So there's our picture of the cell. So these purple little spheres are the phosphate heads. These little yellow things that are dangling off of them are the lipid tails. And so this more brownish color or grayish looking color, that's the fluid outside the cell, kind of the space between individual cells. And then the blue part is more the cytosol, the inner contents of the cell. And so we're going to kind of stay on this slide for a little bit and kind of talk about all these different structures. There are text descriptions of all of them, but I didn't want to spend the time going through all that information visually when we can just kind of talk about it here and go through these different things. So these phosphates are not really connected to one another. They're basically stay in close proximity because of the electrical charges that are associated with both the fats and the lipids as well as with the phosphate. So they're not like glued or linked together in any way. If you imagine like a swimming pool that's filled with just a bunch of like balls, like you might see in like a ball pit, that's kind of what you should think about. If it's just wall to wall, the top of this, like say we have a baby pool that's just filled with all those little ball pit balls, you can poke your hand right through those balls and get to the bottom of the pool. And that's kind of how these plasma membranes are. It's not rigid and really that solid in most of our cells. What's considered kind of a fluid mosaic is often how it's described. Now, in between that phospholipid bilayer, you kind of see these little strings of molecules that are joined together, and those are holding things together internally. So we've kind of got these internal guy wires, so to speak, that are kind of helping give a little bit more rigidity and solid structure to the phospholipid layer, and those are cholesterols. So when, people often think of cholesterol as being a bad thing. A lot of our packaged foods that are sold, advertised, oh, no cholesterol. We need cholesterol. We need cholesterol to hold these cells together. It has other roles too. Now, too much of certain types of cholesterol are bad. They're going to lead to blockages in the arteries, but we definitely need some amount of cholesterol. So that's where, if you eat a no-fat diet, you're really trying to cut calories and you're like, I'm not going to eat any fat. Well, you might run into some problems because we need those fats. We need those fats to make the lipid tails. We need those fats to kind of help make up some of those cholesterols too. So that's kind of the bilayer itself. And then we've got all these other things that are kind of poking through, these blue things and these green things. So we'll talk about those just a little bit. So the green ones here are representing what's called a glycoprotein, or we also have in the back a glycolipid. And so glyco just means sugar, means glucose basically, and lipid is a fat. So a glycolipid is a molecule that's partially sugar and partially fat, and it's embedded in this wall. And you see these little projections coming off of a number of these molecules, and these are what's called the glycocalyx. And it's a way of allowing the cell to be identified. So it's part of our antigen system. So our body needs to be able to recognize cells that are not its own, like an invading bacteria or an invading virus. And it needs to be able to recognize which cells are its own so that we don't mount immune response against our own healthy cells. So that's what these little markers are for. They basically, if another cell comes floating along and bumps up against this cell, the fact that it has these projections, it's kind of like a gang symbol or something. We know that that's our guy. So we're not going to attack him. He's got the red bandana on, so he's cool. He doesn't have the blue bandana, so we're not going to go after him. So that's kind of their key role is as an identifying structure. We've got a ton of proteins that are going to be embedded in these cell walls too. And if you look at this one on the left side, we've got this peripheral protein outside, and then we have the whole blue part that's extending from the outside of the cell all the way into the inside of the cell. And so if a protein goes through the entire phospholipid membrane, it's considered to be an integral protein. It's connecting kind of the inside and outside world of that cell. Some cells only project partially through that membrane, and many will, or some proteins, and many proteins will go all the way through. That peripheral protein on the top oftentimes is either going to cause some sort of enzymatic reaction, or it's a site for something to bind into it, like a receptor site. And that's one way that we control how how the cell is going to decide what it wants to let in and what it wants to keep out. If we look at this blue protein that's kind of sliced down through the middle, you see this little channel going down through the middle. And so this is what's termed a transport protein. if you have a bigger, let's say we have a substance that we want to get into the cell, but it's too big to be able to pass very easily through that phospholipid bilayer, or it doesn't have the right electrical charge that the phospholipid bilayer will let it pass through. We want to have kind of a dedicated way for that thing to get in. So essentially a doorway. And most of these transport proteins, something has to open the door. And so we'll often have a hormone, for example, that might bind to the outside of the cell. And when that hormone binds, that's the signal that tells that channel to open up. There's something out here that's associated with me that we want to get into the cell. And so that's kind of the trigger. And then the same thing can happen on the inside too. If we start developing too much of a certain ion on the inside of the cell, and we need to kind of rebalance things to keep the inside of the cell at the right concentration, then we will have something bind to that peripheral protein on the inside of the cell that will then trigger that gate to open up. And now we can let things flow out of the cell, or actively kind of pump them out of the cell by using some energy. So the proteins that are embedded in that phospholipid bilayer are really, really important for helping control a whole lot of things with the cell. So those are kind of some of the main things that you're going to see in this cell wall. And we'll talk a good bit about some of the ways that things get in and out of the cell and how some of these different proteins are going to help control that. All right, let me skip through these. All right, so transportation was kind of talking about intercellular connections. So we can have structural connections that join adjacent cells together that create linkages. And so those are usually going to be some kind of protein. Proteins are what give our different tissues a more solidness to them and kind of hook things all together. So proteins are really the structural things that hold our body together for the most part. And so that's an important role of some of those proteins. Internally, we have what's called the cytoskeleton. So again, that cyto is the cell, and then skeleton. So when we think about our own skeleton, it's bone. It's very hard and rigid, and that's what gives us shape. And that's kind of what the structures that the proteins that help make up the cytoskeleton do. They're not bone, but they're more just internal supports that kind of either help reinforce the walls of the cell in some way, or they basically link one side of the cell to the other, so that if it gets compressed a little bit, it still kind of holds its shape. It might bend and squish a little bit, but it's not going to completely flatten out. It's going to still, because it has these internal proteins that are somewhat rigid, it's going to kind of help things stay intact a little bit. Enzymes and hormones both are largely protein-based molecules. And so our enzymes are things that are going to help facilitate all of the metabolic reactions in the cell. A lot of times a process might proceed anyways. If we have the right component parts, then cellular metabolism is going to happen. What an enzyme does is it serves as what's called a catalyst. So it just makes that process happen more easily and more efficiently. And so we do need those proteins. And so we're going to have those in the wall to help cause reactions that let stuff in or let stuff out and trigger some of these things. And then even inside the cell, we have a lot of enzymatic reactions that need to take place. We talked about cell-to-cell recognition with the little gang symbols. And so that's that glycocalyx part. Again, has a protein to it, but it also has potentially either carbohydrate and fat, or just carbohydrate and the protein. And then those proteins can also help with transmitting some of the signals. When we have a nerve fire and it's going to try to connect with a given muscle, we have some proteins involved in that process too. So lots of different things that proteins are going to do for us within that cell membrane. Any questions so far? I haven't seen anything in the chat. You guys doing okay? So far, so good. All right, let's keep rolling. All right, let me go back. Hold on, I think I went too far. So that plasma membrane needs to be selective in what it lets in and what it lets out. We want the cell to kind of maintain what it needs and be in charge of that. And we want to also have its ability to kind of block things from coming in just randomly that aren't really invited in. And so those transport proteins are a big part of it. And again, those are usually going to be triggered by some hormone that causes the gate to open or close. How that membrane is put together and the fact that it does have a charge associated with it means that things that, you've probably all heard that like charges repel. So if we have a positive charge trying to get in and the outside of the cell is positively charged, it's not going to let that come in. If we have something that's negatively charged and then it's attracted to an opposite charge, then it's more easy for that thing to come into the cell. So the charge of whatever it is that's trying to get in or out of the cell is going to affect how freely it can pass across that membrane. Concentration gradient is going to affect it. Now, what does that term gradient mean? Anybody? We talk about something have a gradient. What is that?
A gradient would be the intensity diminishes or increases.
Okay, so it's kind of related to the difference between how much there is of something. And so if we have a bigger concentration gradient, it's going to help that thing move more easily than if the gradient is not very different. So if we have, like, let's say we have an ion like sodium. If on the outside of the cell there's 51% sodium and the inside of the cell there's 49% sodium, that's only a 2-point difference. That's not a very big gradient. If I have 80% of the sodium in the area is outside the cell and only 20% is inside the cell, now we've got a 60-point difference or a much bigger gradient. So it's going to be a lot easier for that sodium to want to come into the cell. It wants to reach homeostasis and equilibration where we have a balance, a 50-50 split. And so that the bigger the difference is in that gradient, the more easily it's going to cause things to want to move to kind of create balance between those things. That ionic charge, I already kind of talked about. If things are positively or negatively charged, opposite charges are going to attract and negatives are going to repel. The inside of the cells are generally more negative than the outside of the cell. So positive things kind of always want to come into the cell, and that phospholipid membrane has to be selective about which of those ions they let in. Kim, do you have a question or are you just scratching your head? Okay, I wasn't sure. All right, and then size. If we have things that are really, really small, it's a lot easier for them to kind of sneak in. And so when we look at that phospholipid bilayer, something like oxygen and carbon dioxide, those are small enough molecules that they can just kind of flow right on through. They don't need a dedicated transport passageway. They're just able to slip through the cracks, more or less. If we have something bigger, like a really long chain of protein amino acids that are hooked together, and we've got like 100 of these amino acids hooked together, well, that's too big. That plasma membrane, it's going to bump up against that. It's going to be trying to fit through there. It's like, no, you can't make it through there. You've got a really big dog in your backyard and one of the little fence slats is broken. Well, that dog's going to be able to poke its head out, but it's not really going to be able to escape. But if we have like a little mouse, well, that little mouse is like the oxygen molecule. It's small enough that it can just snake on through that opening and be free. So size is going to affect how easily something is able to pass through or not. Bigger stuff is going to be kept out more easily. We have to have a different way to get that big stuff in. And so we'll talk about that. All right, so when we try to specifically move certain things in or out of the cell, we need to have some way to do it. It can't just sneak through the wall like oxygen or carbon dioxide. We can either move things passively, where we're going to rely on that concentration gradient largely to say, oh, okay, well, we want things to be evened out. So if we have a lot of stuff outside, let's let that stuff in and create some balance. Now, if it's a bigger molecule, we might need to have one of those gates open up to let it come in. But the term passive in passive transport means that it doesn't cost the cell any energy. We're going to let the fact that there is a concentration gradient move those things, because things want to balance out. We want to go from high concentration to low concentration. And so that's just the way it happens in chemistry. And so passive transport, again, we don't need to put in any ATP, and that's our energy currency. When we talk next, we'll talk about active transport. And so active transport means the cell actually has to use up some ATP to move something. And usually with active transport, you're moving things against their concentration gradient. So we might have 80 sodium ions outside the cell and only 20 inside the cell, but we can use active transport to force that 20% out of the cell. And maybe we want that ratio to be 90 to 10 instead of 80 to 20. Because it's like swimming upstream, we're going in the opposite direction of that concentration gradient, it takes more effort for the cell to do that. If we're swimming downstream, essentially allowing that concentration gradient to go from 80 to 20, that's a lot easier. So swimming upstream, swimming downstream, if you think about that, if that's helpful, it's a lot harder to swim upstream. It takes more active energy to go in. So those are kind of the big differences. So simple diffusion is one form of passive transport. Again, this is going to be like our oxygen molecule. When our cells have been doing their thing, using oxygen to create ATP, to facilitate whatever process they're doing. They're making an enzyme, let's say, or something like that. We start creating some waste in the form of carbon dioxide, and that carbon dioxide wants to leave the cell because there's too much of it in there. At the same time, the cell's used up the oxygen it had, and it needs more to keep going. And so the blood that's coming through that area where that cell is going to have more oxygen than the cell, because the cell used up what it has. And so the oxygen is going to want to go from high concentration and leave the bloodstream and get into the cell. The carbon dioxide is at a higher concentration in the cell. And so as that blood flows past, it's like, there's not very much CO2 there. Let's leave. And we can catch a ride on this oxygen, and then we can get carried back to the lungs, and we can blow that carbon dioxide out. So those are both passive processes. We don't need to put any energy into moving those two gases. They're just going to move because the concentration gradients are different. And so that's great for something small. Osmosis is just diffusion of water. So again, we're going to want to cause equilibration. And so if we have a much higher water content inside the cell versus outside the cell, then the water is going to want to leave the cell. If we have a much higher concentration of water outside the cell than inside the cell, then water is going to want to come into the cell. And so this is one reason why you can't drink seawater. Everybody knows, well, you hear about people that are shipwrecked, they're on these rafts floating, and there's all this water all around and not a drop to drink. People sometimes when they're desperate, they're like so thirsty, they're like, I'm just going to drink some of this seawater. And it's really going to dehydrate them faster because essentially that seawater has so much salt in it relative to how much water there is and what those concentrations are in the cell. We drink that seawater, it gets absorbed across our intestine and moves into the bloodstream. Now our blood is really salty relative to the surrounding tissue. And so when that salty blood flows past cells that have some water in it, there's more salts in the surrounding fluid than there is in the cell. And so the water inside the cell wants to try to move towards the saltier area so it can dilute that salt and create equilibration. So it actually is going to pull water out of the cell and dehydrate those cells faster. Even though there is some H2O in that seawater, the fact that there's so much salt in there too is what's going to cause the water to want to dilute the salt, and it'll be pulled out of the cell. If we are really dehydrated, we've been working outside, and we just drink pure water, well, now the opposite thing's going to happen. We've got less water in the cell, more water being introduced into the area, and so we're going to let that water move into the cell more easily to try and create some balance. So osmosis, again, it's usually affected by that salinity, the concentration of sodiums and chlorides and some of the other ions that constitute salts. And that's what's going to draw that fluid across the membrane more easily, or make it leave the cell a little bit more easily. All right, the last two ways that we can move things passively are either with facilitated diffusion or through bulk filtration. And so facilitated diffusion is for bigger stuff. We have those transport proteins that are crossing across that entire membrane barrier, and we're going to have something that triggers that gate to open. It's facilitated in that we need a doorway to let stuff in, and we have to open that door, but we don't need to put any energy into it. And so if we have a higher concentration of something outside the cell than inside the cell, and something triggers that gate to open, now it's very easy for those highly concentrated stuff to follow their diffusion gradient and just flow right into the cell, because now the door is open for them. But we have to do something to open that door. And so that's what that term facilitated refers to. Something needs to help it just be able to follow its concentration gradient, because it's too big to just pass through the phospholipid bilayer, or it's got the wrong charge, so it can't come in because it wants to be repelled away by those phosphates, or the fat, and it won't pass through everything. Bulk filtration is going to use hydrostatic pressure. So that's basically water pressure to move things across the membrane. And so it's if we have like higher blood pressure, then that's going to let things be pushed out of the capillary wall more easily and leave the capillary. So that's kind of an example of how hydrostatic pressure can help things move. I brought this gadget here out to kind of demonstrate that. And this is a filter that is used to filter like stream water or spring water to get all the bad stuff out of it. So it's got this filter on the end, this orange part, and then this part's hollow, and you've got a cap, and then this is just a hollow cylinder. And so you would fill this part with water, and then you can push this in there, and it has this kind of rubber gasket that creates a tight seal. And so then when you push it down, I'm basically creating more force. Now this takes me some energy, and so our body doesn't work quite the same, but I'm pushing pretty hard, and it's kind of moving slowly and compressing down. If I had it on a more stable surface, it would work better. But essentially it's creating enough pressure that it forces the water that's in here to move through the filter. The filter has enough small kind of screening to be able to filter out the bad organisms that might cause infections within us and complications. And so then what's left in here is nice, good, clean water that has been filtered from all the bad stuff from like animal poop that might have washed into the creek. So that's kind of an example of how bulk filtration will work. Again, it's passive in our body, but it's basically just based on pressures in there. So those are the passive ways. So let's talk about active transport. So active just means, again, it's going to cost the cell some energy to do it. But for some reason, we need to do it. And so the most common thing that we're going to do is that uses active transport is the sodium-potassium pump. Sodium and potassium are the two key ions that really cause the difference between the charge inside the cell being more negative than the surrounding outer area of the cell. So we usually talk about most cells having about a negative 70 millivolt charge. So they're 70 points, millivolts below being balanced. And so it's more positive outside, more negative inside. Now, both sodium and potassium are positive ions. And when we activate this sodium-potassium pump, it takes ATP, so we use up a little bit of energy to do it, to basically open this gate, and it's called a pump because we have to actively push these ions against their concentration gradient. So normally the inside of the cell is going to have more potassium, the outside of the cell is going to have more sodium. And so when, let's say like a nerve, if we depolarize the nerve, we activate that, we think about something that we want to move, we send that signal down. Depolarization of a nerve or a muscle causes those charge differences to kind of switch. And so all of a sudden, instead of the inside of that nerve cell being more negative, when it depolarizes, now the inside is more positive. And in order for us to activate that nerve again, we have to reestablish the normal resting membrane potential, or get back to that negative 70 millivolt level. So we need to use this pumping mechanism to now push out the sodium ions that we let in and let back in the potassium ions that we kind of pushed out. And it's a 3-to-2 ratio. So when we activate that sodium potassium pump, it's going to pump 3 sodiums from the inside of the cell and push them back out. Even though there's a lot of sodium already outside the cell, we still want to get those things out there. The potassium that's outside the cell, we're going to let 2 of those back in. So even though they're positive, and both ions are positive, the way that we get back to that negative 70 millivolts is by pumping more positive ions out than the number of positive ions that we let back in. And so again, we're pushing 3 sodiums out, letting 2 potassiums back in. So the net means we've lost 1 positive charge. And so we do that enough times and we can reestablish that normal resting membrane potential. So I'm going to try and play this video. I haven't had great luck playing videos in Zoom, and it's a couple minutes long. It kind of goes through how that sodium potassium pump works, and hopefully you can hear it. Move my mic over closer to my speaker, and we'll see if it's going to work for us. You guys see that?
Active transport is a type of cell transport that requires the input of energy in the form of ATP. The proteins that conduct this form of transport are often called pumps because they force molecules or ions to move from an area of low concentration to an area of high concentration. This is commonly referred to as against the concentration gradient. One of the more common examples of active transport is the sodium-potassium pump. The job of the sodium-potassium pump is to move sodium ions, or Na+, out of the cell, and potassium ions, or K+, into the cell. Let's take a closer look at how this happens. The sodium-potassium pump has binding sites for 3 sodium and 2 potassium ions. Let's follow the movement of the sodium ions first. After 3 sodium ions are positioned within the carrier protein, an ATP molecule is split, releasing phosphate. This phosphate binds to a location on the exterior of the carrier protein, causing the protein to change shape. As the protein changes shape, the 3 sodium ions are released to the other side of the membrane. Next, 2 potassium ions position themselves within the carrier protein, causing it to undergo another change in shape. In the process, the phosphate molecule is released. Once the phosphate molecule is released, the carrier protein expels the potassium ions into the interior of the cell. The carrier then resumes its initial shape, completing the cycle. Notice that for every 3 sodium ions leaving the cell, 2 potassium ions enter. Since both sodium and potassium ions have a positive charge, this unequal movement causes an electrical gradient to develop across the plasma membrane of the cell. A number of cellular processes, including the generation of nerve impulses, use this electrical gradient.
All right, so is that, you guys could hear that? Okay, good, okay, super. So that's just one type. We have a lot of other mechanisms that use this active transport, but that sodium potassium pump is the most common one. And so we're using up some energy. And so ultimately the cell's going to need to replace those ATP and create some more so that it can keep doing whatever other things it needs to do. All right, then we've got bulk transport. So bulk transport refers to a couple of different mechanisms, exocytosis and endocytosis, that are either going to move large things out of the cell in the case of exocytosis, so it's going to exit the cell, And then endocytosis is the way that we get big stuff into the cell. So the EX refers to exiting the cell. You can think about the EN, even though N is IN, I know. That's kind of the way I keep it straight. So it's going to let things in with endocytosis. It's going to let things out with exocytosis. So let's look at those two processes real quick. And both of them are going to use the cell wall to either, if we're having something leave the cell, we basically package it up inside a similar phospholipid bilayer membrane package, and then that package basically hits the cell wall and it merges with it. And so then when it merges with it, then it lets whatever was inside that little package to leave, because it's merged with the wall and it creates an opening where it can flow out. Endocytosis, we've got a couple different ways to do that. So we'll kind of hold off on that description, but essentially it's going to also use the membrane to kind of open up and let something big either be captured or kind of flow into that cell a little bit more easily. But they both use package things up into what are called vesicles. And so that vesicle is kind of just that self-contained little phospholipid bilayer sphere, usually that's going to be merging with the wall or else created from the wall itself if we're letting something in. So here's exocytosis. So let's say this cell was a cell in the pancreas that makes a digestive enzyme that's going to help us break down a fat or a protein in our intestine. So if it's a digestive enzyme, its job is making things that are big and breaking them up into smaller stuff. And so we don't want that digestive enzyme to just flow freely through the inside of that cell, because it has the opportunity to basically destroy anything in its path, because it has the ability to digest these things and break them up. So we want to keep it under control, keep it under wraps. So we're going to package it up after we produce it in this vesicle, so now it can flow through the rest of the cytoplasm and not damage anything until we want it to be released. And so it's going to be packaged in this vesicle. It's going to hit the cell wall. Because the phospholipid bilayer of the vesicle is friendly with the phospholipid bilayer of the cell wall, it's easier for them to kind of blend together and form one boundary. And then we create a little bit of an opening. So now the contents inside that vesicle have a way to escape. And we haven't damaged the cell wall, we haven't damaged anything inside the cell, but now we have released that digestive enzyme, and it can now get into the intestine and help break apart that hamburger that we had for lunch, or whatever the case might be. So that's exocytosis. We're also going to use exocytosis if we have parts of the cell that have kind of died off. Let's say we had a mitochondria, which we'll talk about later. It's been working really hard for a long time. It's done its job. Now it's time to go to cross the rainbow bridge and go to the where all the mitochondria go to die. And so we're going to have some other structures called lysosomes that are going to break that thing apart a little bit and make it into small fragments. And then it's like, well, I don't want this stuff building up in the cell. I don't want my cell to look like a junkyard. I'm kind of OCD. I want it to be nice and tidy. So we're going to create all that old broken down material and send it away because we don't need it. We're going to use what we can, maybe, and recycle, some of those things, but a lot of the stuff might not be great. We're going to do exocytosis if we have captured an invading organism and we have broken it apart. We've given it the death penalty, essentially. We don't want all that bad stuff in there, so we're going to send it back out after we've kind of brought it in and beat it up and broken it apart. So exocytosis is useful for, like I say, getting rid of waste stuff, but also getting rid of products like an enzyme, like a hormone that a cell might be in charge of making. All right, with endocytosis, letting stuff in, we've got kind of three different ways that endocytosis can do. So each of these is a specific type of endocytosis. So phagocytosis, this phage prefix means basically eating, and we've talked about people that can't swallow correctly. That's a dysphagia problem. They can't eat correctly because their muscles aren't working right. The nerves can't activate the muscles to work right. So like stroke patients will have dysphagia, difficult time eating. When we talk about the cell, what's going to happen, we see this phospholipid bilayer in this picture, and the cell is going to essentially recognize, hey, there's something outside that I want to bring in. Maybe it's something harmful. And the most common cells that do phagocytosis are going to be certain types of white blood cells called macrophages. And the phage refers to this, phago here. And so if we have an invading bacteria that's trying to attack our body in some way, we're going to release or try and mobilize these macrophages that then do phagocytosis. So they can extend the cell wall and kind of reach out. So this green thing is the enemy that we're trying to take out, whether it's a bacteria or a virus. And so it's basically, think about like how you might take a bite out of an apple or corn on the cob. You're going to extend your lips and your teeth out of your oral cavity a bit so that we can chomp into it and take a bite. And then we're going to close our lips off to kind of seal it off. And then once it gets on our oral cavity, it's kind of like being in its own little vesicle there. So that's where this term cellular eating relates to phagocytosis. We've got this little mouth that's basically formed by the cell wall. We're reaching out to capture something and encircling it. So now if it is a bacteria, now we've packaged it up in this vesicle, so it's kind of trapped in there, and it can't damage anything else in the cell. And now if we have what's called a lysosome, which we'll talk about in a bit, that has the ability to kind of merge with that, then the enzymes in the lysosome that break things apart are going to be able to start further breaking down that bacteria and negating its ability to continue infecting us. So phagocytosis is often associated with white blood cell activity. There are some other cells that do it, but white blood cells are definitely the most common one that we'll talk about with that. Now, pinocytosis is another way to let things in, and they call this cellular drinking. So if we look at this outer boundary of the wall on this membrane, if you think about taking a glass and you're going to take a drink, but we don't reach out with our mouth to take a drink. We just kind of open things and pour that water in. So we're opening up like our oral cavity right here and letting water and whatever is in that water flow into that space, and then we can seal it off. Just like I can take a swallow and put a mouthful of water in my mouth, close my lips around it to create a little vesicle, and then I can swallow it and go down. So cellular drinking, again, we're letting something come in, but we're not having to extend that cell wall. We're basically causing what's called an invagination, an inward dip in that plasma membrane that lets fluid flow into that space, and then we can seal it off and make this vesicle. So that's a way for us to get in larger volumes of water and whatever else might be like dissolved in that water. So that's pinocytosis. And then the last way that we're going to use endocytosis is receptor-mediated endocytosis. So this is very similar to pinocytosis in that we're going to create an inward fold that the thing can be collected in, but it has to be triggered. So we have to have something bind to a receptor site that says, it's like somebody knocking on your door going, hey, I've got your delivery from Amazon. And you're like, oh, okay, great, my package. I'll open that up so the porch pirates don't get it. If we don't get that knock on the door, we might not know that it's out there, and then it might leave, whether somebody's stealing it or whatever. So we need that signal of something binding to that receptor site to trigger us and say, oh, now's the time to create that little infolding so we can capture whatever it is that's out there that we want to bring in. And that thing was either too big, or it had the wrong charge, or it's not quite able to get through that membrane in some other way. So those are our three ways of letting things in: that phagocytosis, the pinocytosis, and then receptor-mediated endocytosis. All right, pino. I don't know if that just means drinking. I'd have to look it up to see exactly what that is. I'm not sure what the root is. I'm sure it's probably Latin or Greek, but I don't know off the top of my head. You stumped me, Tom. Students often try to do that. I try not to let it happen, but it's been known to happen. So I'm not sure on that one. All right, so let's talk about these different structures. Most of these, if you've had Greek meaning drink, there you go. All right, so I knew it was Greek or Latin. Pretty much everything's Greek or Latin. Dr. Google knew it. All right. So we're going to talk about the cytoplasm, and then we're going to kind of go through all the different organelles and kind of just do a brief synopsis of what those organelles are in charge of. So we kind of have a general understanding of that as we move forward in the class. All right, so the cytoplasm has the more gelatinous liquidy part, the cytosol. It's going to have inclusions, and then it's going to have the organelles themselves. So in the cytosol, we've got a lot of stuff dissolved in there. So we've got like our 3 macronutrients that we consume: proteins, carbohydrates, and fats. And then it has different types of ions: sodiums, potassiums, calciums, all those type of things. Some other small molecules as well that are just kind of there for the cell to use to kind of keep things in balance and help the cell function appropriately. Our inclusions, glycogen is the stored form of glucose, and so both our skeletal muscle and our liver can store extra sugar so that when they need to fuel muscle contraction or we need to keep our blood sugar levels up, we have the ability to do that. Melanin, that helps us get tan or gives us our distinct skin tones, are going to be considered to be an inclusion. And then we've got a few other proteins that are inclusions as well. Again, and they're just there so that the cell can kind of use them when they need them, and they're not contained within any other kind of organelle themselves. All right, so our organelles, we have ones that have their own membrane surrounding them, and then we have some that are just kind of a little bit more freely there in the cell. And so we've got our examples of ones that have membranes kind of listed here, and the ones that don't have membranes that are still considered organelles kind of down below. And we'll kind of go through each of these organelle types briefly to get through there. So our endoplasmic reticulum, that's a mouthful. In this image on the top, we've got the nucleus right there in the middle, and then all surrounding it, we have what's called the rough endoplasmic reticulum. And by rough, it means the outer surface is not smooth. It has these little pebbly bumps on it, and those are called ribosomes. And so ribosomes were some of the non-membrane bound organelles. And so they're attached to this, and a ribosome is a protein factory. So when, let's say, this cell is in charge of making a hormone, which is largely protein-based, those ribosomes on the outside of that endoplasmic reticulum are where we're going to kind of put together the individual amino acids to make a longer protein molecule that's going to serve the purpose of being a part of that hormone that that cell's in charge of making. So again, we'll talk about the rough and the smooth endoplasmic reticulum a little bit. The outer part here is representing the smooth endoplasmic reticulum, so it doesn't have any ribosomes on it. And so if we look down at this little descriptive box on this bottom slide, that's actually showing a transmission electron picture of a endoplasmic reticulum. The rough endoplasmic reticulum makes proteins. The smooth endoplasmic reticulum is in charge of making fats, putting them together, and breaking up carbohydrates. So it's going to help with carbohydrate metabolism as well as fat synthesis. So smooth has a distinct role that's different versus rough. Rough, associate them with the ribosomes, and ribosomes are where we make proteins. The smooth is going to deal with those other 2 macronutrients, either helping synthesize together individual fatty components to make a certain type of fat, or breaking up the carbohydrates so that we can use it for some sort of energy. So those are our 2 endoplasmic reticulums that we've got. All right, let me look at our Golgi apparatus. Some people say Golgi, I say Golgi. I think I'm right. I might not be. Usually you'll say whatever your instructors, the first person that taught you says, and there's some dispute about some of these terms. So if we look at the left edge of this picture, we've got the rough endoplasmic reticulum. So that rough endoplasmic reticulum has been making its protein, whether that's a hormone, whether that's an enzyme, whether it's some structural protein that's going to help give some part of the body a shape or rigidity. And it's kind of produced in not its final form. So it makes this general protein, it floats across this space and is received by the Golgi apparatus, these different layers of green that we're seeing here. And it comes into that Golgi apparatus, and whoever's in charge of production in there goes, well, that's great, but that's not quite the shape we need it to be in. And so it's going to modify that protein, tweak it a little bit, make it so it's just the way it's supposed to be, and then it gets to the shipping dock. And so now this is like the Amazon factory. It's like Amazon accepts stuff from other vendors and then they package it up. Here they're going to package things up. We're going to put them in those vesicles. Even if it's a digestive enzyme or a hormone, we don't want it to act on the cell that produced it, either by dissolving it or causing some reaction that the hormone might cause. So we put it in this protective barrier that then is going to flow through the cytosol, merge with that plasma membrane, and through the process of exocytosis, now we can get the product that was in that package and release it outside the cell, where it can go where it needs to go to cause whatever action it needs to do, whether it's a hormonal response, whether it's a, maybe we made a collagen protein that we need to repair a ligament that got torn, or we're needing to make a digestive enzyme that's going to help us break down something in our GI tract. So that's kind of the role of Golgi, is basically modifying the proteins that the rough endoplasmic reticulum makes and then sending it out the door so that it can do what it's supposed to do. All right, our lysosome, I've mentioned them a little bit. The term lyse just means break apart. And so a lysosome has a lot of enzymes usually that are kind of aggressive, that are allowed to break things up. They act on things that either like dead cell components that we need to break up into their smaller pieces, or an invading organism like a bacteria that we don't want. That lysosome is going to basically merge with that vesicle that brought that bacteria in, and then it can start attacking it and breaking it apart so it can no longer function the way it's trying to cause damage or illness to us. So lysosomes are our friends, usually, unless they're attacking us, which sometimes happens. Very similar to a lysosome is a peroxisome. Peroxisomes use oxygen and basically hydrogen peroxide. If you've ever used hydrogen peroxide, it's going to basically kind of use that action to help break things apart in a similar way that a lysosome does. So it has the same kind of function as a lysosome. It just has, it only does certain things. Lysosome is a little bit more generic. It can kind of do destroy a lot of different things. Peroxisomes can only destroy certain things. And so we have a lot of these in the liver, and the liver is really our primary organ that helps break up and negate things. So if you take Advil, if you take any kind of medications, ultimately we want those medications to be broken down, and the peroxisomes in the liver are one of the primary ways that we kind of help negate that thing. Otherwise, those medications would just stay active forever, and that doesn't happen. We recognize, oh, well, it did what we wanted it to do, but I don't want that thing in my body anymore. It's going to build up and cause problems, so we're going to break it apart. And then I need to take more later if I'm still hurting or still having pain or whatever. So those are both ways to break things up. All right, mitochondria. Mitochondria's nickname is what? Who knows the nickname for the mitochondria? How do we refer to it?
It's the powerhouse of the cell.
Okay, so it's the powerhouse of the cell, meaning it's producing energy. So just like you might think of like a hydroelectric plant or a natural gas plant that's transitioning over that natural gas energy into electricity, the mitochondria are going to use oxygen through the Krebs cycle and the electron transport chain to help generate lots and lots of ATP out of the carbohydrates that we take in, the fats that we take in, and to a lesser extent, the proteins that we take in. So that's kind of its key function. And we'll dive into that, the specifics of that in a later chapter. This is kind of the picture of the mitochondria. It's usually kind of kidney bean shaped. It has a double layer and some inner folds. And the structure is what lets us help generate lots of ATP. So we'll talk about that in more detail in a later chapter. There's our ribosome. We've already kind of talked about the ribosome. That's our protein factories. I'm going to skip through that. And then we've got the cytoskeleton. Again, these are structural proteins that help create shape to the cell, usually. And we've got three different types. We've got microfilaments, intermediate filaments that are a little bit bigger, and then microtubules. So these two filaments are both just solid structures. Think of like twine or something like that, or a rope. A little bit more rigid than that. Microtubules are also going to be long cylindrical things, but they are hollow. They have a hole in the middle of them. And so they're a little bit different shape because they do have that cavity within them. And they each serve kind of distinct roles. Our microfilaments are mostly actin. So actin's a kind of protein. We have a lot of actin in our muscle fibers, but we have them in other cells too. And they just kind of help give a little bit of shape and structure and form to the cell wall. In the case of muscle, then we're going to have that actin is going to interact with the myosin protein to actually cause a change in length and create tension that lets us create movement in the body. So microfilaments. The intermediate filaments are going to help go from cell to cell. So you're going to have them kind of projecting between cell walls to kind of link things together a little bit. You also have them kind of within the cell. If you think about like a spherical cell, they're going to kind of run from wall to wall on the inside to kind of give it support. Think about maybe like tent poles or something like that, kind of giving some shape and structure to the inside of the cell. And then our microtubules, the most common place that we have the microtubules is coming out of the centrosome, and that's related to the process of mitosis as well as meiosis. And those centrosomes are going to create microtubules that kind of grow out of it. And then we'll talk at the end of today about how mitosis kind of proceeds. They're going to latch onto the chromosomes and then kind of pull the chromosomes apart as we're trying to create two new cells. So that's kind of one of the primary roles of these microtubules. Again, some of them also create kind of like little train tracks or highways within the cells so that things that are moving from one organelle to the other kind of have a way to get there. So they also serve in that role too. So we kind of have a more dynamic role where in that process of mitosis, we're going to grow out some microtubules to latch onto something and then kind of reel them in. But they can also be a more static structure that again allows us to be able to more efficiently send things from one part of the cell to the next and cause that. So those are our three kind of cytoskeleton components. And there's our picture. The microfilament, the actin, is this twisted kind of chain of spherical molecules. The microfilament kind of structured a little bit more like a cable. You can see how it's kind of this twisted wrap. And then the microtubule has that hollow opening in it. And that's the biggest of the three. And these two green things here, that's the centrosome. And we'll talk about that. All right, let's look at that. There's our centry. The centrosome is two centrioles. Centrioles are these individual structures. They're usually kind of depicted this way as being kind of at opposite orientation to one another, kind of perpendicular to each other. And that's just kind of the way that they're linked together. And this is a electron microscope slide of how they're oriented. This one's looking at the opening kind of in the center there, and then this one's laying at the opposite angle. And again, their key role is with cell division, and we are going to talk about that at the end here. All right, then we've got our projections on the outside. We've got our cilia and we've got our flagella. Again, the only human cell that has flagella is the sperm cell, and that's that little whip-like tail that helps that sperm cell move and go, up through the uterus, try and get up into the fallopian tubes where it can then fertilize the egg that's been released. Cilia are going to be on the outside. Their role is usually just to try to move things. So we have a lot of cilia lining our different organs that have hollow spaces. So our lungs, we've got those, the trachea, the bronchi and all that, bronchioles, and we've had cilia lining all those things. So when you think about having to clear your throat or having a cough, the cilia help make that more effective. They're going to help sweep that mucus and anything that gets trapped in that mucus kind of back towards the middle so that we get that little tickle in our throat and then we want to cough it out. So it helps us kind of get rid of those things. We have a lot of cilia in things like the digestive tract too to kind of help sweep things along, create mucus, things of that nature too. So kind of any hollow organ that we have in our body is usually going to have some kind of cilia in there to kind of help with moving things along. So here's our picture of the cilia. Again, this is showing a digestive tract kind of picture. Actually, I think it's showing a respiratory tract. So we've got these cells that are producing the mucus, and then the cilia are kind of helping sweep that mucus along. And we usually want it to be in one direction. We don't want that cilia to sweep stuff deeper into our lungs where gunk would build up and it's harder to cough out. So we want it to be sweeping things back towards the middle so then we can hack it out a little bit easier. And then there's our sperm cell with the flagella that they've got shown there. The last little projection on the outside of the cell is the microvilli. And these don't really move. They're kind of shaped like cilia in that they're like little hairs kind of sticking up. But their primary role is to increase surface area. And so we have a lot of these in the intestinal tract that help with absorption. If I have food kind of floating past these microvilli, we have a lot of enzymes in all the little nooks and crannies between those microvilli, and we have a lot of absorptive cells that, so when we have a, say our digestive enzymes have broken up a protein, those protein fragments are going to have a specific passageway that they need to enter, and those microvilli increase the surface area. If that was just a smooth, flat surface, we don't have as much ability to let stuff get in. The fact that we have all these little dips and crevices between those microvilli gives us a better chance to let that protein molecule stick to an enzyme that can break it apart and open the passageway to let us absorb that thing into our bodies. So those are really critical for helping absorption and increasing surface area. All right, last little part inside the cell, we've got the nucleus. The nucleus does have its own little bilayer membrane to it. It has some little distinct pores, and these are basically like the transport proteins are for the cell wall. They're going to let things in or out of the cell. And then in the center, we have the nucleolus. And so that's really kind of the main heart of the cell. And we're going to have our chromosomes inside the nucleus in the form of chromatin or condensed down into chromosomes if the cell's about to split. When we do want to have a cell make a new cell, a new copy of itself, and cause mitosis, then that membrane for the nucleus dissolves. And so when it disappears, now those chromosomes are easier for those centrosomes to kind of latch onto and pull apart, because the membrane's not there kind of blocking their action. So it is usually present, but at certain times in the life of a cell, it's going to dissolve itself and let it have access to the chromosomes that we need to copy and then split apart to create a new cell. All right, the nucleoli are another component that's going to be within the nucleus, and that's where we have RNA. And RNA is basically, that R refers to the ribosome. So these are essentially the blueprints for the factory. So if we need to make more ribosomes, we need to make a new factory that's going to make a protein. In order for the cell to figure out how to do that, it needs to copy the specific section of RNA that's in charge for that type of protein factory. So that's kind of the key thing to know about RNA. We'll talk about gene expression and making proteins later on in more detail, but that's the role of the RNA. So it's similar to DNA, but it's just a single strand instead of a double strand, but it also has the different nucleotides that we associate with DNA as well. So they have a lot of similarities, but they definitely have a different role in the cell. All right, so our DNA is going to be in the nucleus, and it can exist in it. You'll see a couple different terms that describe it: chromatin, chromosomes, chromatid. Those are just different forms of the same thing, basically. Chromatin is how we refer to it when the DNA strand is basically kind of unwound and loose inside that nucleus, inside the membrane of the nucleus. When we're getting ready to make a new cell, we need a little bit more organization than that. We can't just have these loose strands of DNA. It'd be harder to make sure that we got the right mix of things on each new cell. So it condenses down and gets wound up a little tighter to form a chromosome. But it has the same material, it's just different shape. Is it unwound or is it more wound up and condensed? Let's look at how it looks. So this is our, what we call the double helix structure. So this is just one tiny little section of a given strand of DNA or a given strand of a chromosome. We have these nucleotide bases, and we've got 4 of them in DNA, and that's these 4 terms down here: thymine, adenine, cytosine, and guanine. They have a specific way that they're going to pair up, and they just repeat themselves in different orders to create different genetic information. And when we look at human cell or human person's DNA, then we're talking about physical characteristics, eye color, height, things like that are all going to be contained and dictated by this genetic sequence. And when people have defects where things didn't copy over quite right, then we have certain genetic problems, like a kid that might be born with cystic fibrosis. It's something got out of order in that genetic code, and it led to that child developing this problem because now that gene doesn't work the way it's supposed to, and it can't produce the protein the way it's supposed to because it's defective. So we do have a lot of genetic defects that come with problems there. So that's kind of the smallest amplification. If we look over here on the other side of the picture, you can see that same structure. And when we create chromosomes in preparation for cell division, we have to wind it up kind of tight. And so this looks like kind of an old telephone cord. I don't know, some of you might not have ever seen one of those. It's quite possible. Maybe you've seen a picture. But this gave it the ability to kind of extend and retract a little bit if you've never seen one of those. And that's kind of how this looks. We've got these things called histones, and they're basically like little spools. And they help us to kind of further organize that genetic information. So we've got the coil of DNA that will wrap around individual histones, and then all the histones get kind of compressed into another coil, and then that coil ultimately takes the shape of this chromosome. And so this kind of X-type shape with a center kind of joined section, that's kind of how the chromosome is going to exist before we do cell division. All right, how are we doing time-wise? We only got 5 minutes. That's not good. I got a few more slides to go. So let's talk about the life cycle of the cell, and we'll revisit this. I think in the next chapter we go through embryology, so we'll spend more time kind of going into the nitty-gritty of this when we talk about both mitosis and meiosis. Mitosis and meiosis are similar processes, but they have some distinct differences. Meiosis is going to let us have the genetic material, so then like if somebody's going to have kids, half of the man's DNA is going to merge with half of the woman's DNA when fertilization occurs to create a new distinctly individual person. We don't just make a clone of ourselves. Mitosis is making a clone. So it's going to make an exact copy, essentially. If mitosis goes according to plan, this cell that was there splits to become 2 of the exact same cells. So that's a cloning process. Meiosis is not cloning. like mitosis is. So let's real quick go through the phases of the cell. So over here with this G1, all these things in blue are what are called the interphase phases. And so interphase is just kind of the normal everyday life of the cell. That's what most cells spend the majority of their time in these three phases. Well, this slide shows it like maybe like a 60-40 split in time. If we go through that whole cycle, for most cells, it's not. For most cells, this interphase is going to be a much larger portion of the cell's life. The mitotic phase is a brief period of time, but they needed to create room for the pictures, so they made it this kind of split like this. So G1, we've got the S phase, we've got the G2 phase, and then once we complete G2 phase, then that cell is ready to enter mitosis, where we're actually going to create these two new cells. So let's kind of go through these phases real quick, and then we'll kind of wrap up and take a little break before we jump into the lab part. So in G1, we're going to basically, that cell's going to reach maturity. It's going to go about its normal business, make whatever proteins it's in charge of making, if it's an enzyme producing one, if it's producing secretions, if it's producing a hormone, whatever that cell is in charge of doing. If it's a muscle, it's going to do its muscle contraction thing. So that's where it's really kind of functioning and just kind of doing its thing. It's kind of young, it's in the prime of its life. Starts getting a little bit older, and if you think about like a person, well, maybe it's time to I need to start thinking about passing my material on to a new cell. And so it's going to synthesize, and so that's what that S phase is. It's going to make a copy of all its genetic material, all that DNA. And so we're going to have double the DNA that's produced, and that's so when we enter mitosis, ready to be kind of separated, and each new cell is going to have that same genetic material. G2, we're going to create the centrioles and double them so that we have the ability to then produce out those little microtubules that we talked about. They're going to latch on to the chromosomes and help pull them apart. So again, these are all normal parts of the cell's kind of prime of life activities, kind of young and carefree, doing its thing, working really hard, start settling down. I got to think about dividing and leaving something for my offspring. And then G2 are kind of really, okay, that's the nesting period. I got to buy the crib, I got to do all this, so that way when the kid comes, we're all ready to go. So that's kind of our G2 phase. And then the mitotic phase, I'm going to skip through some of these, is when we're actually going to split things apart. Mitosis, prophase, metaphase, anaphase, and telophase. And then once telophase happens, then we actually just kind of have to separate those two cells. So in telophase, they kind of look like, if you've ever heard about Siamese twins, they're kind of joined together still, but with cytokinesis, we're going to complete that separation. So that's not considered a phase. It's just kind of the end of telophase is when cytokinesis happens. Hopefully my internet doesn't give out. I've been getting that message. My wife's teaching in the opposite office next door to us. She's also faculty. All right, let's look at those four phases. Prophase. We're going to condense the genetic material and create what's called this sister chromatid. The chromosome on the left side of that chromatid has all the exact same material as the one in the right. So during that S phase of interphase is when we've created all the copies of that genetic material. Now we're going to condense it down so that way when we start splitting apart, it's more organized and easy for us to kind of latch onto and separate. So I try and associate the order of these four phases of mitosis with kind of an action that's occurring in that phase to make it a little bit easier for students to keep track of. So for prophase, I always think of this as being kind of the preparation phase of mitosis. We've got to get things condensed and organized. So if you think about you're moving, you don't want to just start moving individual items, you're going to put them in boxes and kind of condense them so that it's going to be easier to move and separate from that old living space to the new living space. So we're prepping the genetic material by condensing it down. Metaphase, our chromosomes are going to line up in the middle of the cell. We've got those centrosomes and centrioles that we've replicated on either side, and they're going to grow out their microtubules to latch on to one, each side of the cell is going to latch on to one chromosome, one of the sister chromatids, apart and separate it. And so metaphase, that's when they're going to line up in the middle. We're going to meet in the middle is kind of the term that I think of. So prophase, we're prepping, packaging it up correctly. Metaphase, we're going to meet in the middle for that. We're going to move into anaphase. So anaphase, we're starting to pull them apart. And that's kind of what I associate with the A. We're pulling it apart. So we've prepped, we've met in the middle. Now we're pulling them apart in anaphase. And you can see the sides are starting to kind of pinch in a little bit. And so that's kind of the early part of kind of creating two new distinct cell membranes that are going to surround that genetic material to create the two new cells as we go. So anaphase, we're pulling things apart. Telophase, we kind of further that separation. And I think of like tearing the cell apart is how I associate the TE part. So we're getting this, what's called a cleavage furrow, and it's dipping in closer to the center and getting closer to being able to fully separate those new cells apart. And that's what's going to happen with that cytokinesis. Is going to be the final part of telophase where we actually do separate them and look apart. So the whole reason, or one of the primary reasons we do go through this whole process of mitosis is because the cells, with the exception of neurons, cells have a limited lifespan. And so after they get old and worn out, we need to replace them in order for the whole organ to keep working, the whole tissue to keep working, the whole body to keep working. And so we need this process of mitosis. And different cells are going to have different lifespans. Something like our skin cells or the cells that line our mouth or our gut, those don't live that long. They take a lot of abuse and they're more readily disposable. Something like our muscle cells, they're going to live a lot longer. Now, we still need to replace them as they age, and they will still do mitosis, but they're not going to be as short-lived and as delicate as skin cells or digestive tract cells that are lining it. So cells, with the exception of neurons, pretty much are going to have mitosis at some point, and that's what's going to change the ratio. When we look at that life cycle span, it's going to change how big the interphase portion is relative to the mitosis phase. Something that's has a very short life, like a skin cell, well, maybe it is like that 60 to 40 split. But something like a muscle cell, it's probably going to be more like a 90 to 10 split or something like that, where interphase is a much longer portion of the cell's lifespan, and the mitosis phase represents a much shorter time period with that. Okay, so we got through all that. It's 9:39. I went over by like 5 minutes or so, 4 minutes. So I'm not pretty. That's not too bad. So let's take a little break, and then when we come back, I'm going to set up the breakout rooms. I'll kind of show the different you've been able to pull up. If you don't have Canvas pulled up and you haven't looked at those yet, then I'd suggest maybe trying to pull up Canvas and either look for the announcement I sent out yesterday, and you should be able to open up that PowerPoint link there, or you can go into Module 1 in the course and go into the Lab Activities folder, and it's going to be in that area too. So I'm going to turn off my camera and stuff, up and get something to eat or drink or go to the bathroom, go ahead and do that. And then in, let's say 10 minutes at 9:50, we'll come back in and we'll get going with the lab portion. All right, that's better. So once, if everybody that's present can get their cameras back on, so I know you're here and ready to roll. We'll go ahead and jump into this lab activity. Again, usually when we're actually meeting in person, we do these type of things together in one of our lab spaces. Since we're still kind of having to social distance and do all that. I'm going to see how it goes. We'll try it this way and see how PowerPoint. It's just a set of 5 slides that are basically just pictures, images from the first chapter that we talked about last time. And these are really going to, if you can complete these and you know, kind of recognize all these different structures, these different areas, these different regions, then you're going to be in great shape for the quiz that we'll do next Tuesday when lab finishes up. So that'll be our first quiz next week. So this first one just looks at the different kind of directional terminology that we use. So this is probably the easiest one of the 4. The next slide is probably going to be the hardest one. It has all those different body regions, kind of front and back in the body. And then we look at the different cavities that we've talked about. And then we're looking at those different serous membranes that have the parietal layers and the visceral layers, and then each different organ or organ system kind of has their specific names that are associated with those different membrane layers. And then the final one is looking at kind of the different quadrants and regions in the abdominal pelvic area. And so I've created the breakout rooms. I'm going to open them in a second. And so I made 4 breakout rooms. There's 16 of you guys here in class today. So each breakout room should have 4 people in it. And if you can pull these images up for yourself to view, then you should be able to just, this one doesn't look like it has the lines. Let me find another one like here. So hopefully you're still seeing my screen. If I click on this line right there, it should give me the ability to type in the word. So if you're more old school like me and you wanted to print these out ahead of time and actually have physical hard paper that you can write on, then you could do it that way. But I think you should be able to manipulate these PowerPoints and be able to use them and save this on your computer too to fill it out. So you can do it one of 2 ways. But I'm just basically going to let you guys work in groups of 4 and kind of help each other out in recognizing these directional terms, recognizing those different body region terms, those different cavity terms, and different membrane layer terms, and just kind of spend, I guess, about 45 minutes or so. We've got until 10:40 that lab ends, just trying to work through these things in a group. So you're all coming into the class with different backgrounds as far as how familiar some of these terms are. Definitely feel free to use either your paper copy of the book to kind of help if you've got that handy with you, or potentially using one of the e-versions of the book through Canvas too. I don't know how many computer screens you have available, and so you may want to, I don't know if it's going to be easier to just keep your Zoom open the way you have it if you have it on your computer and still be able to manipulate this image, or some of you may choose to attend the Zoom meeting through your phone so that you have a greater ability to kind of see what you're after on the computer. I'm just not sure what's going to work best for everybody since I haven't done this assignment or this lab activity this way. So if for some reason you want to leave the Zoom meeting on your computer and then re-log in on your phone, I'll pay attention to that and kind of re-let you in. But I'm going to go ahead and open up what's called the breakout rooms. Again, you're going to have 4 people in each room, and it should let you see the 4 people that are in your room if you're looking at the images of the people's faces in Zoom. And then, like I say, just kind of work your way through these documents. I'll kind of take turns popping in and out of the different breakout rooms. I have that ability as the host So I'll be able to pop in there and see if you have questions or other concerns that you're not sure about. But I would suggest trying to do these sheets without looking at a reference and see how far you can get. And then if you need to use the reference to kind of fill in the rest of the blanks, then that would be helpful. But hopefully between the 4 people that are in your group, one of you might have an idea of what those terms are. And then you can kind of use the reference to kind of double check yourself or to fill in things that you're not sure of if you're not sure about some of those terms. Does that sound okay? Sound like we're clear? Okay, I'm gonna go ahead and open up the rooms. And like I say, you definitely want to have this PowerPoint image available and be able to kind of participate with each other and kind of talk through these issues in your small groups. So here we go. kind of letting everybody come back together here. Almost all back in the main room, so to speak. Got a few that are still not returned, it looks like, or a few that may have checked out. I don't know. Now, breakout room 3 is still kind of coming back, it looks like. All right, now we're getting there. All right, I think that's everybody. So I told a couple of the groups, the quiz next week really just uses these images plus maybe a couple others from the textbook itself. So that's going to be your main resource for studying this stuff. Make sure you complete the homework assignment. Has anybody started that or completed that? I haven't checked. Okay, did it go okay? They're kind of busy work, and I admit that. But they do just kind of force you to stay up on things. And I'm sure doing the homework probably is going to help you with the quiz as well. Some of our later quizzes, more of the images will come from the cadaver dissection tool, which I told a group or two. But I'll let you know for sure on that one, which it is. The chapter that we covered today on all the cell, we don't really have a quiz that's specific to that. You will be tested on that knowledge on the written exam. But the second quiz, we're going to start moving into the skeleton and kind of try and shift the focus to some of the more physical therapy specific things that you really need to know more detail about and more information about. So that's kind of where the lecture content is going to become a little bit different than the lab content. So we'll start working through the skeleton. We have a number of extra bedrooms in our house, and I literally have a skeleton in my closet that I'll pull out. And so I've also got individual bones and things like that too. So we'll be using those I'll be using the actual models, and that's where I really wish we were meeting on site. I think it's helpful from a understanding with the three-dimensional components of the bone. That's going to be something that we'll probably miss a little bit in translation with this format versus the actual in-person format. But we'll kind of see how things change as the summer moves on. Since we have fewer than 20 people in this class, we may get permission to come back on site. I know they're trying to really minimize the chance that we're going to spread things, which I am totally in agreement with. So we'll just kind of have to see how that plays out. For sure, next semester you will be on site for at least kinesiology lab, and we'll have those bones there, and we might be able to kind of revisit and pull some of that information back in as we go. How did you feel the breakout rooms worked for this activity? Was it okay? Okay. I think it's good in that it's going to help you guys get to know each other a little bit better, to be in those small groups and kind of interact in that way. So I think there's some benefits to it. So we'll probably kind of keep doing that. We do have handouts like this for labeling and for identifying stuff for a lot of the other activities. So feel free to kind of poke around in some of the modules as we're proceeding through the class. The stuff that's in those modules was all designed for off-site, for actually using it when we're on-site. So I may or may not use everything that's in those folders, but I'll try and give you a heads up once I figure out what I'm doing for each specific lab, which ones might be helpful to download ahead of time if you're somebody that would prefer to do it on paper versus doing it on a slide or something like that. So otherwise, I think we'll call it a day. And be sure and let me know between now and Tuesday if anything comes up, if you have other concerns, other questions. I'm happy to answer those as we go. Okay? But have a good weekend, and I'll talk to everybody next Tuesday, if not sooner. All right? Very good. Take care.